All right. Hi, everybody. I hope that you are all doing well. So, a couple of kind of random things, and also just a couple of general announcements. So, remember that you have the VDJ problems due next Monday. A lot of you have already emailed them to me, but I'm making sure you have plenty of time so that if you want to come talk to me in office hours, you can. There's going to be a flow cytometry set based on what we're going to learn about today. That's going to be also due next week. That's also posted. And we're talking about a primary lit paper on Friday. So all that stuff is available for you on Moodle, so just be aware. The Monday lecture is already on YouTube. Wednesday is not, because apparently the battery in the microphone decided to only last for 40 minutes. And so it's really great for a while, and then it just cuts out. Have to work on that, but it's coming. Today, like I said, I'm going to talk about flow cytometry. But before I do that, just one quick thing. One of the things that I do is I have all of the big journals send me their table of contents every time they publish. And so like a new issue of Nature comes out every Thursday, which is like one of the top journals. So I got the Nature table of contents yesterday, and there was a paper I just want to show you the title of that I thought that is relevant to some stuff we're talking about, and I thought you would be intrigued by, or some of you would be interested in. So just FYI, this came out yesterday. It says that the trash DNA that gets cut out during VDJ recombination can, in some cases, get transcribed and contribute to leukemias. So that was that came in my inbox yesterday. So just thought, since it's relevant to stuff we're talking about, I would tell you. So like I said, today we're going to be talking about flow cytometry. And flow cytometry is a really important technique in immunology that immunologists use a lot. We also use it to describe a lot of our immune cells. And as a result, when I talk about B-cell development next week, I'm probably going to sort of be drawing out flow cytometry data and sort of talking through some flow cytometry data. So I want to tell you about this technique before I actually tell you about B-cell development. And you'll be seeing this technique over and over throughout the semester. The other reason why it's actually kind of interesting to talk about flow cytometry now is that it also falls in the way that most immunologists use it, to being a technique that involves antibodies. And so there are a ton of techniques in biomedical sciences that involve antibodies. And so here it's not about making an antibody to detect a pathogen or to deal with a pathogen. Here we're taking advantage of the fact that antibodies are proteins that are super specific for some other protein. And we're using that property for our advantage, usually for detecting something. And so in flow cytometry, with the way that most immunologists use it, we use antibodies. So it sort of also falls in as one of our antibody techniques that we can think of. And if I had sort of more time to talk about this, I would talk about a bunch of different antibody techniques. So these are just some of the techniques that involve antibodies. And again, this is us taking advantage of their specificity. It's not actually, we're trying to kill a pathogen or something here. So what I want you to just see from here is there's a whole bunch of different assays that take advantage of antibodies. They vary a lot in terms of things like how sensitive, so how much antigen or antibody can be detected. Sometimes we're trying to detect antigen. With these, sometimes it will detect antibodies, usually antigen. So there's a lot of variation. There are a lot of sort of different ways you could do these types of techniques. I'm not going to talk about all of them now, though they are all really interesting. We're instead going to think about a specific problem that we might need to solve as an immunologist. And that is that sometimes what we want to do is we want to have data on particular types of immune cells and how many of them there are and which ones are present. When I have done this course with a lab 1 has actually been about distinguishing different cells under a microscope. This is what some of them look like. And one of the things that students learn in that lab is sometimes it can be a little complicated to tell the difference. You have to definitely, know what you're doing. You have to have some experience to really be able to feel like you can distinguish things well. At one point in that lab, I asked students to go through their blood smears and distinguish 100 leukocytes and count what kind of cell it is, and I tell them to time themselves. And usually it takes them at least 10 minutes, if not longer. And that's to get 100 white blood cells and to distinguish them. And they feel like their data is sort of from that. So this is hard if you're going to do it by eye. And one of the reasons why it's hard is that this is actually a field from a blood smear. All these pink cells are red blood cells, and there's one white blood cell. And this is really kind of what that density really looks like in blood. You can see that there are way more red blood cells than white blood cells. And so first, just finding all the white blood cells can be hard. Different ones are at different frequencies and may be easier or harder to find. But the other thing is that, when, for example, if we were to try to distinguish using microscopy, we would say, look, it's a lymphocyte, the cell of the adaptive immune system. I can't tell if that's a B cell or a T cell. I just know it's a lymphocyte. I can't tell where in the B cell's life it is if it is a B cell. There's a lot that I can't tell there. I just say lymphocyte. And so here's all the different types of lymphocytes. So you can see we've got a bunch of different, T cells versus B cells versus NK cells, and there are different types of T cells, different types of B cells. You can also see another kind of chart sort of trying to break down all the different types of lymphocytes. And so the moral of the story is there's a lot of types of lymphocytes, and you can't even distinguish them at all by eye. And so we have this problem of how the heck are we going to actually start to get good data on what kinds of immune cells we have. And so I want to sort of show you specifically something that I did very frequently in graduate school. It was a problem that was a key part of almost all of my experiments in graduate school. And so in graduate school, I had some mice. I infected those mice with a virus. And I then tried to measure their primary immune response, specifically looking at how many CD8 positive T cells, so one kind of T cell. And I specifically wanted only the ones that responded to one antigen, which was an antigen from HIV. And I wanted to measure their primary response, and I wanted to measure their secondary response when I gave them a second dose of the virus. I have graphs like this that I've published. And you can see I've got the size of the immune response, in this case, the number of CD8 T cells that are specific for HIV at all these different times. And so somehow I had to have a way to know which ones were the CD8 positive T cells that responded to HIV. I took blood from my mice a lot. And one of the things I had to be able to do was tell the difference between the CD8 T cell that responded to HIV and a CD8 T cell that responded to flu or something else. So I had to be able to tell the difference between the CD8 T cells and just measure the ones I wanted. But when I actually took the blood from my mice, this is what I actually would see in terms of all of just the white blood cells. So if I just got the white blood cells out of my mouse, this is actually what I would have. And I even was very into it when I was making this slide, so the proportions are correct here too. And so Somehow I have to be able to count how many of just this one cell there is out of this whole mess of my mouse's blood. And I've got to be able to do it every day. And so this is what I use flow cytometry for. And this is going to be something that we as immunologists do all the time. When we want to measure particular types of cells in an immune organ, in the blood, in wherever, we will use flow cytometry. And so there are a couple of parts of this process that I need to tell you a little bit about. So one of the things I need to tell you a little bit about is sort of general biology of how we tell the cells apart. And then after that, I'm going to tell you about the machine that we use to do the telling apart. So what we actually know is that all of our white blood cells, in fact, probably all of our cell types in the body, but I only care about the white blood cells, so whatever, have special proteins on their cell surface. So it turns out that you can actually identify particular types of cells by what proteins are on their cell surface. So for example, I know that a cell is a T cell because it has a T cell receptor and because it has a protein called CD3 on its surface. I know it's a CD8 T cell because it has T cell stuff and CD8. I can have other kinds of T cells. They still have the T cell stuff, T cell receptor and CD3, but they have something else like CD4. I might call them a CD4 T cell. I can know that neutrophils have a special protein on their surface. I can know that monocytes have special proteins on their surface, and so on. And so you don't need to remember which proteins are on which cell types now. As we move through the semester and we learn about different cell types, we'll talk about their unique proteins. But the idea that I want you to see here is that we have, as immunologists, been able to characterize what are the proteins on different types of cells that sort of distinguish each of our different immune cell types. We're not perfect at it. There's actually a really interesting paper that came out this summer that actually described a new cell type that had not been described before this summer. So like we're still, working some of it out, but we have a basic idea of which sort of characteristic proteins are on the surface of each of our immune cells. And this also brings up one other kind of side thing I have to tell you about. This one always cracks me up a little bit because what I have learned is that this is one of those things where people outside of immunology are like, ugh, can you believe the immunologists do this? It's so terrible. It's the most confusing thing ever. And I'm like, what? It's confusing? I don't even understand how it's confusing. So I want to try to tell it to you in a way that's not confusing. Because apparently some people think it's just terrible. And the thing that they think is just terrible is the way that we're using CD and numbers for some of these proteins. This nomenclature, the CD nomenclature that we use for some of these proteins. And so I want to talk a little bit about how sort of this process of discovering all of this works, and then we can sort of understand what's up with the CD proteins. So let's imagine, and this is really what happened. Once upon a time, Emily was studying her favorite kind of cell. And she found a protein on the surface of her cell. Emily, what do you name that protein?
Emily.
Emily named her protein Emily. Excellent. Nadia also is studying her favorite kind of cell. And Nadia discovered a protein that's on her favorite kind of cell. What do you name your protein?
Nadia.
Nadia. Perfect. And we could go on and we could all have our own protein we discover. So what if Emily's favorite cell type and Nadia's favorite cell type were the same?
Them.
Okay. No. So what if they're the same cell? Is the Nadia protein and the Emily protein the same? Or are they two different proteins?
Hard to say. I feel as though they'd be different, but...
But you don't know. You'd have to figure it out. What if they were the same protein?
Then they are the same.
Then they're the same protein. Then what's the name of the protein?
It has two.
It has two names, right? Like, chaos could happen if we start thinking. So when I think about the characteristic proteins that we have, they're like charts that immunologists often see. I think we're up to at least 500 things on the chart. You could imagine getting some pretty crazy chaos if all 500 of them had like 17 names. right? That would be terrible. In fact, this is a paper that I had to read in my first semester of grad school. And so these authors had to list every name for the protein that had ever been described. So they say the CC chemokine, thymus-derived chemotactic agent 4, so there's one name. Also secondary lymphoid tissue chemokine, there's another name. 6C kine is another name. Exodus 2, there's another name, blah, 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 blah. Like, this is not good, right? Yeah, it does.
It almost misleading. It almost feels like it's those little parentheses is like derivatives.
Yeah, right. It's super confusing, right? I will also tell you, again, as someone who suffered with this in graduate school, still sometimes suffers with this even today doing research. When this sort of thing happens and you do a PubMed search, you usually put in one name. You might do a PubMed search for the Emily protein, and then you would not see any of the papers that called it Nadia. And so then you wouldn't actually know everything about that protein because you'd only be getting half the papers in your PubMed search. So this sucks, right? Like, you understand why this is not okay. So instead, a group of immunologists got together and they made a standardized naming system for these proteins. It's clear that these immunologists must be really fun at parties. But basically, once a year, they get together, they look at all the new proteins people have described on the surface of immune cells, and they figure out which ones are the same and which ones are different, and they give them an official name. And the way the official name would work is that Emily's protein would be named number 1, and Nadia's would be number 2, and Julie's would be number 3, and so on. That's all it is. When they were doing this, they sort of thought that these different proteins had something to do with how the cells were differentiating. They didn't really know exactly what it was. This started in the 1970s. And so they called these the clusters of differentiation, or CD. And so the idea is now immunologists just have this standardized nomenclature for proteins on the surface of cells that are, the first one that was described is now CD1. And the second one is CD2. And like I said, we're up to CD400 or so. I don't remember the whole number. So we have this standardized name for these surface proteins, which is the CD system. And so you're going to hear about CD and some number a lot this semester. And like I said, for some reason, when people are like putting down immunology, they're like, oh, they use this confusing CD system. And I'm like, it's numbers. To make everyone's life easier. It's not that tricky. So you're going to see a lot of use of the CD system throughout this semester. There are some proteins where we still call them by some other name because just historically they have famous other names. Sometimes those names are also really informative, but pretty much every protein does have a CD number. And so we're going to be, so we've got these characteristic proteins on the surface of our white blood cells. Many of them have names with this CD nomenclature. You can see I even have some that I named other things just because the other names are so famous. So for example, B220 most people call it B220, but it's also CD45R, and I could go through the whole list, whatever. You can see that's all the CD thing means. Don't let it scare you. I don't quite understand why it scares people as much as it does. So because we have all of these characteristic proteins on the surface of our white blood cells, what we can do in order to distinguish those white blood cells is use antibodies that bind to those proteins. So we can use antibodies that were made in a lab that bind to different proteins. So we could see I might have an antibody against CD3 that would bind to CD3. I might have an antibody against CD8 that would bind to CD8. Sometimes I might call these antibodies anti-CD3 for the one that binds to CD3. If I'm doing a lot of shorthand on the board, I will write this as alpha CD3. That's often how immunologists write when they mean anti-whatever for an antibody. So I could treat my cells with an antibody against CD3, and it would only bind to the T cells, only bind to the cells that have CD3 on their surface. I could treat my cells with anti-CD8, only the cells that have CD8 on their surface. I mean, that's great, but you still can't actually like see antibodies super well. And so the antibodies that we use in flow cytometry have a modification. They have something added to their FC portion. Here it is shown as a star. And so you can see the little stars on the FC portion of the antibody. And those stars are fluorescent molecules. And so here you can see that my anti-CD3 has blue on it, and my anti-CD8 has red on it. And so I know that a cell that is a CD8 positive T cell is a cell that has blue and red. If it had just blue and no red, maybe it's this other kind of T cell. If it had no blue and no red, it could be somebody else. And so basically what I can do is look at, using these fluorescently labeled antibodies, I can determine what proteins are on the surface of this cell and use that to tell me which kind of cell I'm looking at. So that's awesome, but it still doesn't completely solve our problem of wanting to do this. Because what I haven't told you about is, how do we actually look at the blues and the reds? How do we actually look at the cell and count how many cells have blue and red? How many cells have blue only? How many cells have red only? How many cells have nothing? Like, how do we do that? You could imagine that we could do it with a microscope, right? So I could look at a microscope, I could look at what color each of my cells are. I could like make a little chart in my notebook and count. It would still take forever to do like 100 cells. You would still have a little bit of error. Like, there would still be some issues there, right? And so instead, we've come up with a machine that helps us solve this problem. And so immunologists all use these machines to help us distinguish different types of cells. And this machine is called a flow cytometer. And so we're going to talk a little bit about what's going on inside the flow cytometer and what the data look like and sort of how we interpret those data. So the first thing that we do if we're going to do a flow cytometry experiment is we take our cells. And the cells that we're going to use here are usually a mixed population of cells. This is a heterogeneous population of cells. You got all the cells in the blood that came out of the blood of your mouse, or that came out of whatever your sample is. You're going to take that heterogeneous population of cells and you're going to add the antibodies of interest. So in the experiment I just showed you, you would add anti-CD3 with blue and anti-CD8 with red. I think those were the colors that we're going with the right ones. So I would add the antibodies and let the antibodies bind to the proteins on the cell surface. So then I'd have a whole mixture of cells. Some of them would have, 2 antibodies, some would have 1, some would have 0. And you can see there's my mixture of cells. So I have my whole mixture of cells. I'm then going to take my mixture of cells in a tube and put it in the machine. And so you can see this is basically showing that whole set of cells in the machine. You can see I've got some cells with 2 antibodies, some with 1, some with none, got some free antibody. I got a whole mixture of stuff there. And I'm going to mix all of this in a whole bunch of liquid, which is kind of what you're seeing up here at the top. And then the machine is going to use a bunch of different physics principles. And yes, in grad school, I had to learn all the details of all the physics of this. And it was actually, the person who taught it was so bad at teaching it that he kept comparing every part of the machine to a radio. And I'm pretty sure I left knowing no more about flow cytometry than I knew before, but I knew how to build a radio. That. But the moral of the story is you take all these cells in all this liquid, and you use some physics principles to actually force them into a stream where those they're going to basically be coming in little droplets here. You can see the droplets of all this liquid with cells coming here. You actually, in old machines, it was actually a vertical drop with gravity assist, where you're going to be sort of having this work out to drop the cells. But what's cool about this dropping, the thing that's really important about the dropping, is that when you set up your machine, when you start your experiment, you do some adjustments so that your machine is dropping 1 cell per droplet. So what you can see is you've got these little droplets with 1 cell per droplet falling out of the machine. Yeah, the one that I have doesn't actually use gravity anymore, but that's fine. But imagine it falling. Falling is fine. So we got 1 cell per droplet falling. So it's sort of individual cells that we can identify. And that does take some sort of interesting machine setup. There are also some interesting ways that we have to look at how we look at our data to make sure we don't have, sometimes the machine messes up and has 2 cells per droplet or 2 cells stuck together. And you do have to do some work to deal with that, which I'm not going to get into now. I'm just going to say, imagine it's 1 cell per droplet. Don't worry about how you do the controls for that. Yes?
So how small are these droplets then?
It's basically like a drop of water, or like a drop, yeah. Yeah. So what you should imagine is that the amount of liquid that the cells are mixed with up here is in vast excess. You have a lot more liquid than you do cells. And that's not drawn exactly right here, but this is the best they can do. Drawing's hard, yeah.
You have to have a proper proportion of liquid to cells that make different places for concentration.
The machine is putting in this liquid that's labeled here as sheath fluid, which you don't care about, in such excess that you don't really have to. But one thing I will tell you is that ideally you're looking at sort of individual cells. They're not crazy sticky. They're not clumps of cells. There are some cell types that don't do well in flow cytometry. If you think about blood cells, blood generally moves through your body. Blood cells are not supposed to stick to other things, right? Some cells of your body, like if you tried to put skin cells through a cytometer, it would be all sorts of bad because the skin cells like to stick together. And so they'd try to stick together and you'd probably clog your whole machine up. There are different types of cells. I've run some crazy kinds of cells. In general, if a cell is sticky or like not particularly round, you got to do a whole lot of special stuff. There's a lot of sort of control that goes into that side that I'm not going to get into now, though if somebody really wants to think about the physics of it, we can. The moral of the story is we're dropping 1 cell per droplet. We should hope that they're not very sticky. For example, when I was in grad school, we had somebody who was looking at immune cells in breast milk, and so she would run breast milk through the cytometer, and there are a lot of like other cell types that slough off there, and I would like arrange my entire schedule just so I didn't have to be after her, because the machine would be so gross and cloggy afterwards. So we've got our 1 cell per droplet dropping, and those droplets are going to go into the path of a laser. And so we're going to have a laser. In most modern cytometers, we have multiple lasers instead of just one, but the laser is fine for right now. And we're going to look at how the light of that laser is changed by interacting with the cell and by interacting with any fluorescent molecules that were on those antibodies. So we're going to get some information about sort of the size and shape of the cell and sort of some physical properties of the cell based on how it interacts with the light. And also we're going to get some information about what antibody colors are stuck to it because of how they interact with the light. And so basically we're going to shine a laser on our cell, and then we're going to detect the light that comes off. And depending on the cytometer, we might have different numbers of detectors, depending on how fancy your cytometer is. So like the cytometer I have here lets me read 5 things about the cell. The cytometer I use in grad school let me read 20. There are cytometers out there that go above 50. So it's just sort of a question of how many detectors you've got. And then that information from the detector goes into a computer, and you're going to use it. Your cells go into the trash. So basically, they go straight into waste at this point. So you basically analyze what was in that population, but you aren't going to have the cells back afterwards. And so here is another view of all of that. Hopefully, this maybe works volume-wise. You never know.
Flow cytometry is a powerful technique for the analysis of multiple parameters of individual cells within heterogeneous populations. Flow cytometers are used in a range of applications, from immunophenotyping to ploidy analysis to cell counting and GFP expression analysis. The flow cytometer performs this analysis by passing thousands of cells per second through a laser beam and capturing the light that emerges from each cell as it passes through. The data gathered can be analyzed statistically by flow cytometry software to report cellular characteristics such as size, complexity, phenotype, and health. In this tutorial, we will look at how a flow cytometer works, how scattered light and fluorescence are detected by a flow cytometer, and how the resulting data can be analyzed. This view shows the primary systems of the flow cytometer schematically. These are the fluidic system, which presents samples to the interrogation point and takes away the waste. The lasers, which are the light source for scatter and fluorescence. The optics, which gather and direct the light. The detectors, which receive the light. And the electronics and the peripheral computer system, which convert the signals from the detectors into digital data and perform the necessary analyses. The interrogation point is the heart of the system. This is where the laser and the sample intersect, and the optics collect the resulting scatter and fluorescence. First, let's talk about how the sample is delivered to the laser. Here we see how the sample is transported through the interrogation point. For accurate data collection, it is important that particles or cells are passed through the laser beam one at a time. Most flow cytometers accomplish this by injecting the sample stream containing the cells into a flowing stream of sheet fluid or saline solution. As you can see, the sample stream becomes compressed to roughly 1 cell in diameter. This is called hydrodynamic focusing. In fact, flow cytometers can accommodate cells that span roughly 3 orders of magnitude in size. In most cases, cytometers will be detecting cells between 1 and 15 microns in diameter, although through the use of specialized systems, it is possible to detect particles outside this range.
So that's sort of some information that you can see here about what's going on with the cytometer. One thing I forgot to mention when I was here is so I mentioned, for example, to you that in the lab, when I've done labs of this course, I've had people count and identify 100 white blood cells, and it usually took them 10 plus minutes. It just took them a while, and they felt like their data was eh. And you couldn't tell which kind of lymphocyte was which kind of lymphocyte. Here, when I was in grad school, I would run my samples for 2 minutes each, and I would often maybe get 100,000. Sometimes there are situations where you can get up to like 1 million cells in that amount of time. And you're getting way more data about them because you're getting the amount of proteins as well as some other parameters about the cells. Yep, Jazz.
Do the machines or do the flow cytometers doing more developers really they grab more fragments of the optics that went in?
So basically they're looking at light coming off in different wavelengths. So they're looking at more different wavelengths of light coming off. So for example, if I added antibodies that were red and blue, I would need a detector that can detect reds and a detector that can detect blues. So that would be 2 detectors. If I had 20 detectors, they would be detecting 20 different colors. And so I could have 20 different antibodies, each with a different color, and I could start to really kind of distinguish who's who of those cells.
Okay.
Does that make sense?
Yeah.
Okay. Yeah. So we need to think a little bit about exactly what we are seeing going on with this cytometer. So here is my droplet. Here's my cell. Here's my laser that I'm shining on it. There are a couple different things that happen in addition to what's going on with the colors in my answer to Jazz. So one of the things is that when you shine a light on it, and you could imagine, if you don't believe me that this is a thing, you can sit in a dark room and shine a flashlight at like a ball or something. You would see the exact same thing. One thing that happens is the light kind of gets bent around it, right? This is known in flow cytometry as forward scatter. And forward scatter actually ends up being a measurement of how big the cell is. So if it was a tiny cell, the light would get bent around it less. And if it was a ginormous cell, the light would get bent around it more. So first thing I get to know about every one of my cells, every single million of my cells, is how big is it, based on how much the light bends around it with this forward scatter. And this is, again, independent of we haven't even got to like the color part of the laser yet. The other thing that we get to know is a little bit more, like really getting into the detail involves a little bit more physics, and I'm not going to go there. I've always explained it in a sort of funky way, and once one of my physics colleagues was around, and she then like explained the physics of it, and I was like, wow, that was so much better. But I'm not going to do that right now. But the other thing that happens is the other way that our cells vary is based on what's inside of them. So some light is going to go through the cell and not sort of get deflected around it. Some of the light's actually going to make it through the cell. And if there was like nothing in the cell, the light would basically go through the cell. If there's a lot of stuff in the cell, particularly a lot of granules, sometimes also lots of internal complexity, but really imagine lots of granules. When the light goes through the cell, it's going to get bounced around. And it's going to keep bouncing off of things. And it's probably going to come out at a weird angle. Again, a physicist could explain this so much better than I could. But really what you need to know is, did the stuff inside the cell bounce the light off of it? Like bounce the light weirdly. And so we actually can find out how many granules are in the cell, or how much stuff is in the cytoplasm, by looking at how much this light is bent from that stuff. And this is known as the side scatter. Which we usually say is a measure of granularity, while forward scatter is a measure of size. And so we're going to get forward and side scatter, and we're going to get a measurement of size and a measure of granularity for every cell. This is like single cell. Like I could get a list of every million cells and get what was their forward scatter value and what was their side scatter value. Like I could have a table if I wanted. I wouldn't want a table of a million cells and that information, but I could if I could have it if I wanted it. So,
Flow cytometry is a powerful technique.
This is the one I wanted.
Now let's see how laser light is used to detect individual cells in the stream. As a cell passes through the laser, it will refract or scatter light at all angles. Forward scatter, or low-angle light scatter, is the amount of light that's scattered in the forward direction as laser light strikes the cell. The magnitude of forward scatter is roughly proportional to the size of the cell, and this data can be used to quantify that parameter. But how can we record this scattered light? Light is quantified by a detector that converts intensity into voltage. In most cytometers, a blocking bar, called an obscuration bar, is placed in front of the forward scatter detector. The obscuration bar prevents any of the intense laser light from reaching the detector. As a cell crosses the laser, light is scattered around the obscuration bar and is collected by the detector. Because small cells produce a small amount of forward scatter and large cells produce a large amount of forward scatter, the magnitude of the voltage pulse recorded for each cell is proportional to the cell size. If we plot a histogram of these data, smaller cells appear toward the left and larger cells appear toward the right. A histogram of forward scatter data is a graphical representation of the size distribution within the population, but such a graph only presents one-dimensional data. Next, let's look at side scatter. As we have already seen, a cell traveling through the laser beam will scatter light at all angles. Light scattering at larger angles, for example to the side, is caused by granularity and structural complexity inside the cell. This side scattered light is focused through a lens system and is collected by a separate detector, usually located 90 degrees from the laser's path. The signals collected by the side scatter detector can be plotted on one-dimensional histograms, like we saw for forward scatter. The one-dimensional histograms we have seen so far do not necessarily show the complexity of the cell populations. For example, what appears to be a single population in the forward scatter histogram is, in reality, multiple populations that can only be discerned by looking at the data in a second dimension. This is done through the use of two-dimensional dot or scatter plots. You can see that the peaks from the forward and side scatter histograms correlate with the colored dots in the scatter plot. Now we can view the results obtained when we create a scatter plot using forward and side scatter data from a typical peripheral blood cell run. The populations that emerge include lymphocytes, which are small cells possessing low internal complexity, monocytes, which are medium-sized cells with slightly more internal complexity, and neutrophils and other granulocytes, which are large cells that have a lot of internal complexity. This multi-parametric analysis is the real power of flow cytometry. Now let's take a look at another parameter.
Oh, I don't want to do that one right now. Oh, good. I stopped it there. Good job. These are actually data from when I was in graduate school. And so basically what you can see is that what the machine does is you get a value for forward scatter, side scatter, as well as all of the how much red, how much green, how much whatever for every single cell. And then you can make graphs of that or plots of that. And so here we have how much forward scatter each cell had. Here we have how much side scatter each cell had. And you can see every dot on here is a cell. And so like I said, I think this is like 200,000 cells. And so you can see, this cell is pretty small and also not very granular because it's relatively small on forward scatter and it's pretty close to the bottom on side scatter. This cell is like really big and medium granularity. A cell up there would be high granularity. And you can even see that the cells tend to kind of come into clusters. This is what blood looks like on a cytometer. You get this one cluster of cells that are sort of not very granular. They're also relatively small and have like not that much complexity inside them. There's my lymphocytes. I can get some cells that are bigger and sort of are a little bit more internally complex. There's my monocytes. I can get some cells that are way granular, this big burst at the top. Those are my granulocytes. And so I can just take some blood, throw it on the cytometer, and I can get these amounts, these sort of distinctions really easily, way more easily than I would do if I actually was trying to distinguish them under a scope. One thing that you can also notice about this is sort of what very classic cytometry looks like. We don't have to show the data like this. They showed you in the little video that you could just have a histogram of like just forward scatter, and you could see like there's a lot of cells here and there's fewer cells here or something like that by just having cell number or some other parameter on this graph. So we can do the dots like this, but you can also look at this and be like, and if I asked you, I'm gonna ask a very, very challenging question. All right, ready, Jazz? All right, see this spot right here? And see this spot right here?
Correct.
Which one has more cells?
That top one over there.
This spot right there?
Maybe now.
How do you know? This spot and this spot.
Because it's just a bigger and darker spot.
Okay, but I mean the actual spot that I'm touching with my finger.
Well, I don't
That spot and this spot. Why do you know?
Because it's hard to differentiate amongst like, clusters.
Yeah, at some point, like, it's just like all black and you can't tell the density, right? So people have come up with other ways that you can basically show the exact same thing. So sometimes people will show it as like contours of the density of cells in different locations. And so you could see like, this is where the top of the mountain is. And so that was the most dense kind of thing. And sometimes people do the actual dots with colors so that you get like density and individual dots. This is definitely my preference, but you can see lots of different ways you could do this. But the idea is just that you're trying to see which cells are where in some of these different views. So our laser also is going to let us look at fluorescence. And so if you recall, our antibody has this molecule on its Fc portion that is a sort of a label. Officially, it's a fluorophore. But we're shining the laser light on that molecule, and as a result, light comes off of it. And we actually can know some things about, like, if I shine this kind of light of this wavelength on, I get this kind of light off. You don't have to really think about different wavelengths. You can just know their colors. I shine a particular color of light on, I get a particular color of light off. And I can know for each different fluorophore what color I get off.
One of the most common ways to study cellular characteristics using flow cytometry involves the use of fluorescent molecules, such as fluorophore-labeled antibodies. In these experiments, the labeled antibody is added to the cell sample. The antibody then binds to a specific molecule on the cell surface or inside the cell. Finally, when laser light of the right wavelength strikes the fluorophore, a fluorescent signal is emitted and detected by the flow cytometer. How is this fluorescence information collected? The fluorescent light, coming from labeled cells as they pass through the laser, travels along the same path as the side scatter signal. As the light travels along this path, it is directed through a series of filters and mirrors so that particular wavelength ranges are delivered to the appropriate detectors. Fluorescence data is collected in generally the same way as forward and side scatter data. In a population of labeled cells, some will be brighter than others. The voltage pulses are recorded and can be presented graphically. What if we want to do a two-color experiment? If we analyze data from the two-color experiment using a scatter plot, 4 distinct populations emerge. Looking at the dot plot in terms of quadrants, cells with only bright orange fluorescence appear in the upper left quadrant. Cells with only green fluorescence appear in the lower right quadrant. Cells with both bright green and bright orange fluorescence appear in the upper right quadrant. And finally, cells with both low green and low orange fluorescence appear in the lower left quadrant. Multiple fluorescence parameters are necessary to dissect complex biological systems.
So what you can see is that in that video, we had cells that had different amounts of green and different amounts of orange, right? Some of them had no green, some of them had no orange, some of them had like 1 green, some of them had like 4 greens, right? So the amount of green varied and the amount of orange varied. And what you saw in the plot is that you could plot the amount of green and amount of orange for each cell. And again, you could sort of distinguish between your different cells. You, as the experimenter, picked what was on each axis. So you picked, I'm going to look at how much green versus how much orange. You can actually pick whatever you want. Again, in the machine that I have here, I can do forward scatter, side scatter, and 3 colors. In grad school, I did forward scatter, side scatter, and 18 colors. And so you can get a lot of information about each individual cell. So for me right now, I can have 100,000 cells, and I can have 5 measurements about each of those cells, forward scatter, side scatter, and how much of 3 colors are on that cell. You could imagine other cytometers will give you a lot more.
One of the most common ways to
I just
study
watched
cell-
that. So like these are actually 4 of the colors that a lot of people use pretty often, sort of and so you can see basically what we're seeing is like how much green, how much yellow, how much orange, how much red is on each of my cells. And so this is sort of what this ends up looking like, is that we have our mixed population of cells. They're both structurally different, and they have different antibodies that have bound to them based on what proteins are on their cell surface. Those antibodies have different colored fluorescent molecules on their Fc portion. They're going to go through a stream, one by one, individual cells. We're going to shine a laser, get 2 parameters about the size and other physical properties of those cells, as well as however many colors data we have on the cytometer we have. And all that information is going to go to our computer, and the cells are going to go in the trash can. And so we will get data that might look like this. And so here you can see, again, this is data from my graduate work. So here you can see I was looking at the CD3 that had 1 color. So really I was using anti-CD3 to see how much CD3 was on the surface of the cells. Some of them had very little CD3. Some of them had a lot. So I can distinguish like high, like I can actually have like a continuous variable. It's not just yes, no, it can be like high, medium, low, can get amounts. I can also get the amounts of CD8s. I could, if I wanted to, just look at CD3 versus cell number as a histogram. And so here, if I did that, you'd see some cells have a bunch of CD3 and some cells don't have any. CD8, I could be like, some cells have a bunch of CD8, some cells don't have any. Or I can do this 2-dimensional dot plot where I look for each individual dot at each cell. How much CD3 did it have? How much CD8 did it have? And you notice I kind of get 4 populations, some that have a bunch of CD3 but no CD8. So this would be CD3 positive, CD8 minus. Some that have neither, so this would be CD3 minus, CD8 minus. Some that have just CD8, CD8 plus, CD3 minus. And some that have both, CD8 plus, CD3 minus. I can't remember right now which question it is on the problem set, but there's one where I just want you to label the 4 quadrants with the plus and the minus and tell me like, this one is CD8 plus CD3 plus. This one is CD8 plus CD3 minus. That's all I'm looking. I redid the problem set, so the questions are in a different order than they used to be, so I don't know what number it is right now. But that's all I'm asking on that one. People sometimes get way too into that. So this is cool, but there are some additional things that you can do with your software. So one of the things that you can do with your software is you can make little regions. And those regions are called gates. And so in these data, I drew 3 gates. So you can see I drew this pink circle. I said, I care about the cells that are in this area. I care about the cells that are in this area. I care about the cells that are in this area. I drew these 3 regions or gates. And when I do that, the software tells me, okay, cool. This is what % of all the cells in your sample were in that gate. So now I can look at the sample and I can know in, a minute, oh, it had 60.7% lymphocytes in that blood. And it had 3.81% monocytes and 28.4% granulocytes. And so you can get this information. And so being able to draw gates is really key. But there's one other part about gates that is also really key. If you think about it, I've told you on like a fancy cytometer, you might have 18 colors and forward scatter and side scatter. I'm only showing you graphs of either 1 dimension, 1 parameter with a histogram, or with 2, CD3 and CD8, right? If you try to imagine a graph with like 4 axes, that would be in like 4 dimensions, and your brain can't actually like imagine 4 dimensions, right? Or 20 dimensions or things like that. But what you can do is you can start by gating. So you can say, okay, these are my lymphocytes, these are my monocytes, these are my granulocytes. I know that because I'm an immunologist. And then when I do this, I can double click on the lymphocytes and say, okay, computer, just show me the cells that were in that gate. And now I'm going to look at 2 other colors on them. And then I might just pick some that I like there and look at 2 other colors on them. And so I can keep subsetting the cells more and more and more. And so here is an example. And so if I gate on cells, I would say that I was gating on the lymphocytes. That means I'm looking at only the lymphocytes. I'm ignoring everybody else. I'm eliminating everybody who's not in the gate for future analysis. It doesn't mean I'm like throwing the data away. It's still in the computer. I'm just telling the computer only show me this stuff. And so for example, I could take my lymphocytes, my monocytes, and my granulocytes, and I could say I want to see each of them by themselves, how much CD8 and how much CD3 are on those cells. And so you can see that, you know, in the lymphocytes, I've got these 4 different populations. In the monocytes, I really only have mostly this one population. The other ones aren't very common. Granulocytes, same kind of thing. And I could get numbers, percents on these as well. And so I could then further gate it and further gate it and further gate it using all of my different data. So I can get a lot of information off of these data. This is showing you sort of another way of thinking about this. So here what you can see is they have decided they like this group of cells for whatever reason. This is from like a flow cytometry tutorial online, and I have no recollection of what the cells are, so I have no idea why they picked those. But they picked those. They were 24.9% of the original sample. And they said, I only care about those. So they clicked on those, and they could then look at them with CD8, which is on the y-axis, and CD3. And so you can see that here, and they can say, I really like cells that are both CD3 positive and CD8, which are, you can see here are sort of the blue ones. Those are 7.1%. And I want to know of those cells, how much of CD45RA do they have? So I could double click on those, and then basically I could do a histogram, or I could do a two-dimensional thing if I wanted to as well. And so here I can see, you know, plus or minus for this. Here again, I picked, oh, I like this population of cells in this situation. I can do plus or minus for this other marker on them. And so I can keep sort of subsetting and keep getting into different information about the cells. And I'm always getting things like percents and numbers in my computer. So then I can take whatever the data is that I get and, you know, graph it or something like that. We're going to see flow cytometry data a lot this semester. And so these are some examples of flow cytometry data here. These are actually 2 different views of the same general plot. And I'm also going to draw a version on the board. So, so this is actually a very standard plot. You will know this plot later in the semester. This is what happens when you run cells from a thymus in certain conditions. This is just what you always get. I laugh at this because when I was in grad school, when we were second years, you had to design a t-shirt for like the whole program, because there was like this event, and the second years designed the t-shirt. And every year the second years would like spend all this time like designing their t-shirt. And then this one professor who was really good at art would be like, yeah, no. And he would redraw, and he would draw something. And so my class was like, we're not putting effort into this, because we know whatever we make is going to get thrown away. So we're like, we're going to kind of be jerks. We're going to use this. So we actually just gave him this. Sometimes it's known as the thymus bird, or at least we used to refer to it as the thymus bird, because it looked like a bird. He loved it. He thought it was the best t-shirt idea he'd ever seen. And so I have a t-shirt somewhere with the thymus bird. But my grad school class still kind of thinks of the thymus bird as our mascot. So this is the thymus bird. And these are just 2 versions of it. So what you can see is that we've got CD4 on one axis and CD8 on one axis. You decide when you're doing the experiment what's on which axis. So, for example, if I tell you, to draw me a CD4, CD8 plot on an exam, you could do this. You could also do this. I don't care. As long as the cells match in the right place, I don't care how you made the axes. But you should draw me some axes. What you can also see is that oftentimes people will put gates on their cytometry data that look like what you see in the middle. Officially, this is called making quadrant gates. And so basically you have this cross that you put in. You don't have to do it as a cross. You can draw shapes. So you saw some data I did before where I was drawing shapes. Oftentimes, when I talk about data with you guys, I'm going to just talk about it with simple quadrant gates. And so you can see where they took this sort of data and they made quadrant gates. And so they did something like this. They made 4 quadrants, okay? And we can talk about what is the name of the cells in each of these quadrants. So what are we going to call the cells in this quadrant? Yeah, Emily. Okay. Sometimes, especially when we're talking about thymuses, we call this the double negative because it's minus for both. Who's here? Yeah, Jazz.
CD8 plus and CD4 minus.
Okay. Sometimes we call this like the CD8 single positives. Who's up here? Yep, Jazz.
CD8 plus and CD4 plus.
Okay, and sometimes we call those the double positives. And then who's this? I'm trying to see if there's someone else. Yes, Brooke. So we've got CD4 positive, CD8 negative. Sometimes we would call those the CD4 single positives. You could see we could do the same thing over here. We put the axes different, but this is still the same cells down there. Still the same cells up here. The only difference is now the CD4 single positives are down here, and the CD8 single positives are up here. Okay, so do you understand how to sort of read and think about the plot like this? All right, awesome. David.
So you mentioned gating. This is this is for fluorescence from the antibody. For this thymus gland data, for the size, is it all just very similar?
So usually when you look at a thymus, it's relatively similar in terms of forward and side scatter. You get mostly lymphocytes on a thymus. And so, yes, you can draw gates on that too. You can draw gates in lots of different ways. I just want you to sort of be able to read some of the data here. Yep, Genesis.
So for the first and fourth plots,
Mm-hmm.
obviously like positive and negative, and then there's also
Yeah, because it's whatever this is positive, because here we're, minus of CD8, plus of CD8. You could, if you wanted to say low of CD8, high of CD8, I would also accept that. Here you can say high or low. And so basically it's just, is it high, is it plus or minus for each of the things in each box? Or is it high or low of each of the things in each box, okay? All right, somebody else had a question. Emily, somebody? Okay, perfect. So we can see the data like this. There's one other thing we have to think about in terms of drawing the data. There's one other thing that I care a lot about when you are drawing data. And so I'm going to draw this plot the way I would want you to draw the plot. I guess I don't actually need this one, but we're going to draw this plot over here, okay? What you can notice in this plot, as you can see from this, is that there are different percentages of cells in each quadrant. There's a different amount of cells in each quadrant. And so if I was asking you to draw this plot for me, I would want you to draw it in such a way where we could tell sort of relative differences in the quadrants. It doesn't have to be perfect, but I want it to be relative. And so the way I would draw this plot is like this. I have a very small population in this quadrant. I have a very large population in this quadrant, and I have medium-sized populations in those two quadrants. I am not really going to care that you knew the difference between those two, but you need to know when we get to the T cells, when we get to thymus, for example, that this one is really big, this one has a lot, this one has a little. So really what I'm going to be asking you to do is think about data sort of plus minus high low in terms of quadrants, and just think about the population of cells in that quadrant. Is there a lot or a little? And you should depict that with a big circle or a small circle. And so that drawing that I have on the board is how I would draw this, and how you might see it like on an exam if I was asking you questions about it or something. So that corresponds to these kinds of plots. And so in the end, basically what I did in grad school is every day I would take blood from my mouse. I would add some antibodies that had different fluorescent molecules. I would make a graph. I would find, for example, on this particular day, there was 3.05% of cells that were CD8 positive T cells that also bound to my antigen of interest. Yes, I'm not telling you how I measured my antigen of interest. Just say, I measured my antigen of interest with a fluorescent thingy. So I get 3.05% of cells are in the gate, and then I put that on a graph. I would like type it into an Excel spreadsheet and then get a graph that looks like this. That's how I did all of those types of data. And so we're going to be looking at different types of B cells, and we're going to be thinking about kind of what we see at different stages of development, and we're going to be seeing data that either is a flow cytometry plot or is like a graph that came from flow cytometry throughout the semester. I've got one more slide about one other application of flow cytometry. And for some people in the world, this is the most important aspect of flow cytometry. For one thing that was sort of implied in the videos you saw was that flow cytometry started out as a just immunologists thing, but now a lot of biomedical scientists use it. And so people use it for molecular biology things to measure aspects of DNA. People use it for cell biology things. Some people use it for like measuring plankton in a water sample. Like lots of people use it for lots of different types of things. And so it's not just our machine anymore, lots of people's machine. But there is a modified version of the machine. Because I'm an immunologist, I get a little cranky about nomenclature. I don't actually care that you are correct about this nomenclature, but I have to tell it to you correctly. Because there is also this, like I said, this modified version of the flow cytometer. If you looked at all the versions of the flow cytometer that I've shown you up until now, the sample goes in the trash can. Remember, after we measure it, we learn some things about what was in there, and then it goes in the trash. But there's a way you can have a machine, these machines are super expensive, that don't put the cells back in the trash. There's a way that you can do it where you actually get the cells back, and then you can do another experiment with them. And what's really key about this is that this machine will actually sort the cells for you. So for example, you can see the computer, I'm running my cytometry, I say, oh, I have some A positive, B positive cells. I have some A positive, B minus. I have some A minus, B minus. I have some A minus, B plus. I got these 4 kinds of cells in my mixture. I really want just cells that have both A and B on them. I would like to do experiments on this kind of cell and not have all this other business contaminating and making my life difficult. And so there are cytometers that will actually sort for you. So that they will actually basically have the droplet, read what's going on with the droplet, know what, is that droplet up here? Is that droplet in the good area or is it not in the good area? And then put that droplet in a specific tube, whether it's a good drop cell or a bad. The way that that works is actually right here. So here you can see the drop, here you can see the fluorescence. What's also happening when that droplet is dropped is it's actually getting an electrical charge added to it. So basically the droplets all get a charge. Let's say it's a plus charge. I don't remember what charge it is. They get a charge. And then they're falling with the charge. There are some plates. And what happens is we go to the computer decides, yes, we like this cell, no, we don't like this cell. If yes, the plates maybe become one kind of charge to make the droplet fall one direction and go into one kind of tube. And if we don't like it, the plates flip to the other charge and shoot the droplet in the other direction into the other kind of tube. And so you can actually separate or sort your cells. And so here you can see, it's not really showing the charge going onto the droplet, but there is one. Then we've got these plates that are flipping charges to divert the droplets in different directions. And so we actually sort the cells and then we can use them for future experiments. And so the cells are not going into the trash can at this point. They're actually something that we can take back out and use. Yep.
In order to sort these cells, you would already have to have like a criteria for them, right?
You would already have to have a criteria. Yeah, you would have to, you set up your sorter and you tell it what you want to sort. This, of course, as you can see from this name, is called sorting. I've said the word sorting a bunch of times. And if I'm being a technical immunologist, I would call the machine that does this a fluorescence activated cell sorter, FACS. Some people who are not picky immunologists use FACS or FACS for all of flow cytometry. I tend to be picky and say flow cytometry if you're just analyzing and throwing the cells in the trash, and FACS is when you're sorting. FACS is when you have a cell sorter. I definitely cannot afford to have a sorter here at Drew, so I do not do FACS, I just do flow cytometry.
I searched it up. If I had to call it something, I'd call it expensive. That's what I'd call it.
It is definitely expensive, but we have one more thing that is a great last minute of class thing to talk about, because Jazz said what he would call it, and he would call it expensive, and yeah, it's crazy expensive. We can have a conversation about how much what I have in the lab costs, which is just a low, simple one. And if you want to actually watch sorting, this is a little video that shows it. But this is the best part. So you can see this machine is called the fluorescence activated cell sorter. FACS is the what we usually use as the abbreviation for it. What does it sound like I'm saying when I say that? Sounds like I'm saying FAX, like facts, right? Yeah, so it turns out that this machine was discovered the year before the fax machine. And when Xerox made up the fax machine, why did they call it a fax machine? Sending stuff. You're sending a copy of something, right? You're sending a copy of something. And in fact, what they said back in the late 70s was that you were sending a facsimile of something. And so they're like, we're going to call it a fax machine because it's a facsimile. And they went to go get their patent on their fax machine, FACS. Because see, they went to go get their patent on their fax machine. And they were told, nope, somebody already has a patent on a fax machine. You can't have fax for your patent. And they were like, well, crap. But our name is Xerox, and that has some X's in it. So we can't use this, so let's use this instead. And so the reason why the fax machine, that's now old and outdated, is called a fax machine with an X is actually because this machine was discovered the year before. And so that was our fun fact for the day, for the rest of the weekend. You have the flow cytometry problem set, which you should know the stuff for addressing now, and I will see you guys on Monday.